So in our last discussion, we gave you a somewhat detailed definition of homology, and we gave you some examples and looked at how we map those onto phylogenies. And hopefully you realize now that we need a phylogeny, that it's only in the context of the phylogeny that we can establish homology or decide if it's something like convergence, which is a form of homoplasy. We need to understand that phylogeny. And in later chapters we'll be looking at and practicing and talking about how we establish these phylogenies and find the historical relationships of species. But for now, you should be able to look at a phylogeny, look and see if a character occurs twice and we have to map it independently, that would be a homoplasy, in this case convergence. If we map it a single time and everyone inherits it, that's a synapomorphy, which is a form of homology. Okay, so again, quick review. And then we looked at how this plays out at the molecular level, and we looked at the cow and the monkey, the lysozyme example, and we talked a little bit about the complexity, but we're going to make it even a little bit more complex here as we move forward. And we decided that yes, these molecules are homologous, but remember their ability to digest cellulose was gained independently, so it was convergent. And so that gives you some more subtleties, just like a forelimb of a bat and a bird can be homologous at the level of the forelimb, but convergent in the fact that they gain the ability to fly independently. Molecules can show similar complexity. So the lysozyme is homologous as an enzyme, but it gained the ability to digest cellulose independently in these two lineages. And the only reason we know that is because we mapped that molecule, the origin of it, which occurred in the ancestor of all mammals, and then the ability to digest cellulose mapped independently in two different places. And again, for fairly simple patterns, mapping is fairly straightforward, and you can do it just kind of intuitively by drawing lines across branches where the changes occurred, and then looking to see whether it makes more sense to have them occur once or many, many times. Now, the process of assessing this homology is especially critical for molecular evolution. And the reason is because we have a somewhat simpler character than we do with morphology. With morphology, we've got all of this complexity in many different shapes and sizes and location, and look at the fine histology or cellular level of things. So we have many, many different ways to evaluate complexity. And so it might be a little bit more of a fruitful purpose to look at all of the details. But when you're looking at genes, all you have are 4 base pairs. And although they can be in many, many different orders, that complexity is much less and can get covered up much more easily as mutations occur because we only have those 4 basic molecules to look at. So let's look at this morphological example of, the molecular example of the lysozyme, and let's complicate it a little bit. So it turns out that the Hoatzin, which is a very interesting bird. This bird occurs in the Amazon and the Orinoco river drainage, so Brazil, Colombia, Venezuela, is interesting for a number of reasons. One of which is that their offspring have claws on their wings when they're juveniles, which is an ancestral trait, and this is one of the only birds that has that trait, although they lose them in the adults. Right. So anyway, they're interesting for a number of reasons. But one of the interesting things about them is Hoatzins also have a lysozyme that allows them to digest cellulose. And most birds can't do that. So it's a unique feature amongst the birds, just like amongst the langur monkeys and relatives and the cows and relatives. They're unique amongst the mammals in those two instances, those two convergence. So here we have a third one with a very similar enzyme. And so we're going to try and establish, is this homologous? Now, it turns out that when you look at the Hoatzin enzyme, it's actually more closely related to another group of lysozymes that originated as a gene duplication event. And so we're further complicating this idea of homology. So to make it clear for you, and this is important to understand because at the molecular level, this happens all the time. Let's walk through this process. So I've got a diagram here where we're going to look at 3 species, species A, B, and C, all derived from a single common ancestor. And then we have 2 speciation events. So B is more closely related to species C, and then A is their more distant relative. And the black lines here represent the species relationships, kind of the big fat tree. The colored lines inside that represent the history of genes. And we're not looking at individual alleles like we were with that earlier diagram. So this is just one gene that's become fixed in this population. All individuals carry that gene. Okay, and it's represented here ancestrally by the green line, that ancestral gene. Now, anciently in this example, in the ancestor, there was a gene duplication event. So this is a mutation that duplicates the gene. So you have 2 copies, and eventually that spreads through the whole population. So soon every individual in this population carries 2 copies of this gene. And this is a very common mechanism. In fact, it's the most common mechanism for where new genes come from. And so we now have 2 copies of this gene represented by the green line and the red line. When the species breaks up, so here we have a node representing a speciation event, right? That's the common ancestor that then breaks into descendant species. Each of those descendant species carries 2 copies of that gene represented by the green line and the red line. And so another speciation event occurs, descendants carry 2 copies of the gene. So the outcome of this, when we come in modern days, we sample these organisms and we find 6 copies of this gene, represented here by the numbers 1 through 6. So, are these 6 genes homologous? So think back to your definition of homology. Similarity due to a common ancestor. And so the short answer is yes, all 6 of these genes are homologous. We could line them up, we could compare them, we'd find some similarities. And although mutations and changes may have occurred along these lineages, they're most likely still very similar to one another, maybe even functionally similar, and can all be put into an alignment. So let's talk about some of the physiological and evolutionary outcomes of a gene duplication event. Originally, when the duplication first occurs, we have redundancy, right? So this gene is going to be the exact same function as this gene originally. Later it might mutate, and then selection and genetic drift might work on it to drive it to become distinct and different. But initially, the outcome of a gene duplication event is redundancy. And that might even provide an advantage. Just having 2 copies of a gene could be useful. You could make more of it more quickly because you could express both copies. There's an evolutionary advantage also in the long run, that having extra copies might provide more flexibility in the way mutation and natural selection and genetic drift could drive the evolution of the genes and create new function. And so often there is even some selection for this duplicated gene once it occurs. But for whatever reason, you end up with 2 copies of it. Okay? So, although all of these 6 genes are homologous, and we wouldn't call these alleles of one another, right? An allele would be if you had different versions of the same gene, one that gave you blue eyes, one that gave you brown eyes. But when you end up with 2 copies of it, that's not an allele, it's just a new gene, right? Closely related and ancestrally connected to the original gene. And in fact, it'd be hard to say which one's the original and which one's the new one because they're just both duplicated versions of that original gene. Okay? So gene duplication leads to extra copies of the gene. Now, even though all 6 of these are homologous to one another, realize that they are related to one another to these ancestral connections in different ways. So notice if I look at gene 4 and gene 5 and I trace where they connect to one another, they connect here at this speciation event. And the reason that species B and species C both have this gene 4 and 5, both have these similar genes, is because they inherited them when the species broke up. But if I look at, say, 4 and 6, I get to this area and their ancestor still has what we would call the ancestor of gene 4 and the ancestor of gene 6. Here, same thing. Ancestor of gene 4 right there, ancestor of gene 6 right there. And they don't connect back into each other until the gene duplication event. So 4 and 6 are connected to one another and share a common ancestral gene, but it's a much more ancient ancestor than the connection between 4 and 5. So although these are all forms of homology, they are different. And so we're going to give them different names, and we'll look at those names here and define them in just a little bit. But realize that there might be different types of homology that are possible depending on the evolutionary history of these genes. And sometimes genes are lost. If there's a new selective environment, they're no longer needed, or just a random mutation occurs and it can be fixed sometimes even if it's not an advantage. But so this pattern can occur also. And so in this case, if we lined up all 5 of these genes and this individual seed didn't have it, we might not know the ancestral pattern. So realize, even though I'm showing you what happened historically here, in reality, all we have when we come along is the data itself, these gene sequences. And we have to be able to figure out these ancestral patterns from the data in our modern times. So it can be quite complex. And there's one other wrinkle I'm going to throw in here. So we might not know ahead of time until we did our analysis whether gene 5 here was more closely related to 4 or more closely related to 3. So here's the next one. There can also be, although this is rare in eukaryotes, it does happen, and it's quite common in prokaryotes, there can also be horizontal gene transfer events, where a gene from one species gets moved into another, maybe a close relative or maybe even a more distant relative. But a gene gets moved into another one. And so here we have a new gene, right? This gene was lost, but gene number 5 somewhere along the line was transferred into an ancestor of species B. And so now species B has 3 copies of that gene. Species C only has 1 copy because of the gene loss, and species A has 2 copies. And so untangling, figuring out the connections between all these genes can be quite complex. Now you've been given this diagram, and so you know, because you can look at each of the ancestors and what they carried, where new events occurred, whether a loss or a horizontal gene transfer event or a gene duplication event, you know the pattern of and the history of these genes. So given a diagram like this, you should be able to trace back where those ancestral connections are. And now let's give you terminology for 3 different types of molecular homology. So these are all subcategories of homology at the molecular level. The first one, which is kind of what we think of as the standard form of homology, it's a subcategory that we are going to call orthology. And orthology means that we can trace common ancestry back to speciation events. So gene number 1 and gene number 3 are orthologous copies of the same gene because they connect back here to that same ancestral gene that splits up when the species split up. That is orthology. Paralogy is homology also, but it's homology that can be traced back to a gene duplication event. So let's look at 1 and 2, or I could do 1 and 4, or any number of these. 1 and 5 would work also. But 1 and 2, we trace back at the speciation event, they're still separate. So there's the ancestor of gene 2, there's the ancestor of gene 1, and they're not connected, they're not coalesced into a single gene copy, they're still separate. But as we go farther back, they finally coalesce and descend from that same common gene in the ancestor, and they split up at the duplication event. So genes that trace their common ancestry back to a gene duplication event are defined as homologous via paralogy. Again, a subcategory of homology. And then finally, if genes can be traced, so gene 6 and gene 4 have to trace back through this horizontal gene transfer event. So if there's a horizontal gene transfer event back before we get to whatever the connection is, then they are defined as xenologous. Xenos is the Greek root word meaning alien or foreign. So they're transferred over from a foreign or a different species. So these are all forms of homology. Xenology is fairly rare amongst the eukaryotes, but it does occur and there are many good examples of it. But it's quite common through horizontal gene transfer of the bacteria. Now notice, if I selected different combinations of these descendant genes to try and understand the phylogeny of the species, I might get a misleading answer. And we'll talk about that in much more detail, but paralogous copies of genes is one of the things that can make inferring phylogeny of species somewhat difficult. And also if xenology is common, that can make the phylogeny, understanding the history of those species, difficult. So most of the time we assume that the gene history matches the species history. And if we don't have gene duplication events, if we don't have horizontal gene transfer events, then that's normally true. There's one little subcategory called lineage sorting, which we'll get to at a later date. But most of the time, if we correctly reconstruct the history of genes, then we know the history of those species. However, paralogy needs to be sorted out. If there are horizontal gene transfer events, those need to be sorted out. And if those events are rare, then sorting them out is not too much of a problem. And we'll look at some of the ways that's done, again, in later chapters. But if gene duplication is very, very common and gene loss is common, if horizontal gene transfers are common, then suddenly using gene histories to infer species histories becomes much, much more complicated and difficult. Okay, so know these definitions, be able to look at a diagram such as this and be able to determine whether two genes are homologous via orthology, like 2 and 4, right, because they trace their ancestry there. 5 and 2, also orthologous because they connect back at the speciation event. Or if we have paralogous copies. So 5 and 1 are paralogous copies because they don't connect back into each other. 1 traces its history back down the green line. 5 traces its history back down the red line, and they don't get back together until the gene duplication event. Or if they're xenologous, if I do 6 and compare them to any of these genes, they're going to be xenologous because they have to be transferred back through a horizontal gene transfer event. Okay? Now, it turns out that because the Hoatzin lysozyme is a different version of this lysozyme gene because of a duplication event, that we still have 3 separate instances of convergence, once in the langur among the primates, once in the cow, and then once in this paralogous version, right? So all of these genes are paralogous because they're connected through gene duplication events. Okay? And so it just kind of illustrates how we can evaluate and look at the complexity and really, really come to fine terms to understand the history of gene evolution and how we gain certain features. The last thing we're going to do on this lecture is introduce this very, well, I was going to say simple, but it's actually somewhat complex. We're going to introduce this idea about sequence alignment and how we accomplish sequence alignment. If the sequences have not diverged very far, if they're very, very similar, then usually sequence alignment is trivial. It's easy to do. However, the more distantly genes are related, the more mutations that occur, and the more different they become, the more difficult this might be. So let's look at 2 DNA sequences and how we might align them. So ATG, CGT, CGTT. And then we have another sequence. Now, obviously, there's been either an insertion or deletion because the sequences are of different length. But the idea is, where do we put that insertion deletion? How do we line them up? So notice at the beginning it's easy. A matches A, T matches T, G matches G, C, G, T, C, G. It's easy. And then we either put a insertion deletion event here, or we put it there and put the move that T over one place. Now in alignments, insertion deletion events are represented with a dash representing a missing base pair in one or more sequences. So in the previous discussion, we showed you a alignment that was missing 3 nucleotides, and we had represented those as dashes in one of the sequences. Okay? So to do this, one of the very common ways, and we'll look at it in more detail in our next discussion, but one of the common ways to do this is an algorithm where we make a matrix like this. And I put one sequence along the x-axis, the other sequence along the y-axis, and then everywhere they match, if they are both are T's, we put a dot. And then the idea is we want to traverse this matrix from one corner, the beginning, all the way to the other one, and we want to get to the end somehow. And so notice that there are 2 different pathways where we could do this. In one of the pathways, we just go straight and we align every dot and every line connecting dots represents going from 1 to the 2nd to the 3rd to the 4th to the 5th to the 6th position in that alignment. And so notice that if we align them this way, we go all the way just straight to the end, and then we just put a gap at the very end to represent where moving from here to there, and that movement laterally represents a gap. But notice there's a second way to do this, that we could potentially go A, T, G, C, but then we could put in a gap, and this would be 1, 2, 3 base pairs to get to this next CGT alignment. So notice there's a CGT here, and then there's a CGT there. So I could put a gap here that would go ATG, gap, 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 and then the CGT here could line up with the CGT there. And then we need to put additional gaps on the end if this is the end. So that's probably not as good of an alignment because we have to infer more evolutionary events. And a large insertion deletion here, and then another insertion deletion here at the end. And so we would tend to prefer this first alignment that we only have to put in one gap. Now, if there were no gaps at all, if there are only substitutions, then this is an easy thing to traverse all the way from one corner to the other corner. But if they're base pairs, then we have to start moving either up and down or left and right to represent gaps in the alignment. Okay? So if we have only substitutions, it's a very trivial thing to do. But if there are insertions and deletions, and as we get more and more of those mutational events, it becomes more and more complex. Okay? So here is probably the best alignment, and we would put a little dash here at the end representing the insertion deletion event that corresponds to that thymine in sequence 1. This is called a dot plot and is an algorithmic way to find the very best alignment between two sequences. Now, there are 3 different events that we need to consider in any alignment. And we'll define these. We'll give them variables to implement them into an algorithm. You're not actually going to be doing those algorithms, but I just want you to understand these 3. The first is the cost of a substitution, right? And this is moving kitty corner through a place where there is no gap, no dot. So for instance, if that was an A at that position and this was still a G, we wouldn't put a dot there. We would still move directly across that, but we would represent that as a substitution event. Now in this alignment, it's fairly simple. There are no substitution events. But if there's no dot and we just traverse it from one corner to the next, that would be the cost of a substitution. The next thing we have is the cost of a gap, or what we would call the gap penalty, which is a W. And this would be represented in our dot plot by moving either vertically or horizontally to get to a new sequence of similarities. So in this alignment number 2, we have a W, or a cost to move over here. But notice in this one, we don't just move, there's just not one base pair, but we actually move over 3 base pairs. So this is not only a cost of a gap, but also making the gap larger than just one base pair. And so we use the variable S to represent the cost of a substitution. We use the variable W to represent putting in a gap. And then we use this variable G to represent a gap that's bigger than one base pair. And we could implement these mathematically to come up with a way to traverse this dot plot in the least costly manner. That's the idea. So notice as we go across here, we don't infer any costs until the end, and then all we would have would be the cost of a gap with no extension of that gap. So depending on what values you give these variables, you might get different answers. But in this case, this is probably, almost certainly, is the least costly way to do an alignment, because we can align everything together and then just put a single gap penalty at the end. That's that G. But here we have a gap penalty there plus an extension, right? I'm sorry, the gap penalty is W here. A gap penalty W times 3, because it's 1, 2, 3 that we have to move it across. So whatever the extension penalty is, that G, that would be times that. And then at the end, if we want them to align all up, we have to put in more gaps because we have to traverse over all the way over here to get to the end. Now, if you didn't follow this exactly, you're not going to be looking at gap penalties. I'm not going to make you calculate the cost, but just realize this is an algorithm that allows us to decide between competing alignments and come up with different costs or different values for those alignments. And that way we might be able to decide whether alignment A is better than alignment B or alignment C. There might be multiple alignments, but we can come up with a very, very best way to do that using the lowest cost to traverse that dot plot. Now, we'll get into a little bit more dot plot. I'll reiterate it and give you a reinforcement of that. And then we'll look at how we do it if we have more than 2 sequences. What if we're aligning 3, 4, 5, 30, 40 different sequences? How do we figure out the very, very best alignment? And that will be a subject for our next discussion.